Okay, so now let's move on to active transport, which is going to require either directly or indirectly some sort of energy source. For the sake of AMP1, our energy source is going to be ATP, or adenosine triphosphate. So everything that we talked about previously, so simple diffusion, the two types of facilitated diffusion and osmosis, the molecules or the water are moving down their concentration gradient, and they do not require any sort of energy. Active transport is going to require energy, and a lot of times this is because we are moving substances against their concentration gradient. So from an area of low solid concentration to an area of high solid concentration. So we have two major active membrane transport processes, and this is kind of confusing because one type of active transport is called active transport. The other type of active transport that requires energy is vesicular transport. Again, both are going to require ATP in order to move the solids across the plasma membrane, and a couple of reasons why they can require energy is if the solute is too big for the channels, if the solute is not lipid soluble, or like I said, if we're trying to move the solute against its concentration gradient. So active transport is always going to require some sort of carrier protein, at which point whatever it is allowing to cross is going to bind to that carrier protein very specifically, but also can bind reversibly. When we talk about carrier proteins and transport and movement of substances across the membrane, we have two types of carrier proteins. One is an antiporter and one's a symporter. And antiporters are going to move substances in opposite directions. So if two substances are moving, one's moving to the inside of the cell while the other one's moving to the outside of the cell. Or we can have symporters in which both of the molecules are moving in the same direction, either both into the cell or both out of the cell. And either way, we're trying to move these solids against their concentration gradient from an area of low concentration to an area of high concentration, and this is going to require energy in order for it to do. So we have two types of active transport. We have primary active transport, which is going to directly hydrolyze ATP, and then we have secondary active transport, which is going to be driven by the energy that's actually stored in a concentration gradient that was created using energy to begin with. So it requires energy indirectly. So again, if we're talking about primary active transport, the energy that's providing that's allowing the molecule to cross is going to come from ATP, which is adenosine triphosphate, and it's going to be hydrolyzed, not the entire ATP molecule, just the phosphate. So if we're looking at the structure of ATP, it's called adenosine triphosphate because it has 3 phosphates. So what happens is when we use ATP as an energy source, we hydrolyze this terminal bond between the 2 phosphates here, and we attach this phosphate to whatever channel or whatever substance that we need to supply energy to. And once we supply that phosphate to provide the energy, we're left with ADP, which is called adenosine diphosphate because now we only have 2 phosphates. The most important example that you're going to see again several times in AMP1 and then again several times in AMP2 is the sodium-potassium pump. It's really, really important. It pumps 3 sodium out of the cell for every 2 potassium that it pumps into the cell. And it's in all plasma membranes of every cell. Why do we need a pump to pump 3 sodiums out and 2 potassiums back in? Because our plasma membranes also have leakage channels that constantly allow sodium and potassium to leak down their concentration gradient. So we have to have that pump that requires energy to push the sodium and potassium against their concentration gradient. And this is going to allow the cells to maintain an electrochemical gradient, which is really important for our excitable cells like our neurons and our muscle cells in order for them to function correctly. So here's a picture of our sodium potassium pump here in purple. We are pushing 3 sodium out. So sodium is represented by these little yellow balls for every 2 potassium we bring in. If you look at this picture, we see that we have a really high concentration of sodium in the extracellular fluid. So we see 1, 2, 3, 4, 5, 6 sodium out here. We only see a couple sodium inside. This pump is putting more sodium in the extracellular fluid. So that's moving it against its concentration gradient from an area of low concentration where we only have 2 to an area of high concentration where we have 6. Anytime you move something against the concentration gradient, it's going to require energy. So we're going to use our ATP right here. We can see that our terminal phosphate was used to provide the energy for the sodium to bind. So the phosphate right here from ATP is bound, and we're left with adenosine diphosphate down here. So it's going to pump 3 sodium out. It's going to then release them and allow for 2 potassium to come in and bind here. If we look at the concentration gradients for potassium, we see we have very little potassium outside in the extracellular fluid, but we have a ton in the intracellular fluid. So again, we're moving them against their concentration gradient, which is going to require energy. So secondary active transport, again, it requires energy, but it requires energy indirectly, and it's also called co-transport. So basically, here's our sodium potassium pump right here, where we're pumping 3 sodium out and bringing 2 potassium into the cell. So this requires energy. What happens in secondary active transport is that this high concentration of sodium on the outside of the cell can be used to bring other substances into the cell. So what happens is glucose, or molecules like glucose, they basically piggyback off of the high concentration of sodium and piggyback off of it and enter the cell with sodium right here. So we can see we have a sodium coming in here, we have a glucose coming in and piggybacking off of sodium already entering and going in. We don't see any ATP here because this does not require energy when we move glucose along with sodium. However, it requires energy indirectly. So if we didn't have the sodium potassium pump over here creating this high concentration of sodium out here in the extracellular space, we wouldn't be able to bring glucose across to begin with on the inside of the cell. So that's kind of secondary active transport. Our second major type of active transport is vesicular transport, and this is what we're talking about when we actually move really large particles, macromolecules, even something like a bacterial cell. Anything like that is going to require a vesicle, which is going to require energy. We have 3 types of vesicular transport. We have endocytosis. So endo means that it's going to move something into the cell. We have exocytosis, so we're moving substances out of the cell in that case. And we also have vesicular trafficking, where we move substances within the cell from one area to another. So say we're moving it from the nucleus to the ribosome or something like that. So endocytosis, you do not need to worry about the clathrin-coated pit or anything like that. Just have a general understanding. Endocytosis, we're bringing something into the cell. And how do we do that? We basically pinch off or invaginate the plasma membrane right here. And eventually it's going to pinch off to form this vesicle right here. One specific type of endocytosis is phagocytosis. And again, you don't need to worry about the clathrin coating or the receptors or anything like that. The big thing here is phagocytosis is going to be used if we're talking about like actually eating a particle, so some sort of solid debris or even an entire bacterial cell. And what happens in the case of phagocytosis, where we bring in this large particle right here, once we pinch this off, we form what's called a phagosome. So a lot of cells are capable of this. So things like macrophages are capable of phagocytosis, and some white blood cells. So for example, our neutrophils are also capable of this. Pinocytosis is different from phagocytosis in that we're not actually trying to bring in a particle, a bacterial cell, or some sort of debris. Instead, we're bringing in a little bit of the fluid from the environment that the cell is in. So we say that it's cell drinking, not really cell eating. So the plasma membrane enfolds and takes in a really small amount of fluid, and once it pinches off and forms the vesicle, we call that an endosome, not a phagosome. And the whole purpose of this is to sample the environment. So our cells are constantly doing this, bringing in a little fluid and checking basically what the status of the extracellular fluid is. The third type of endocytosis is receptor-mediated cytosis, and this is very specific type of endocytosis and transcytosis. That's really meant for macromolecules, so things like insulin and other hormones, or even unwanted things can enter this route, something like the flu, and they have to come in and bind to a receptor in order for it to cause this invagination and vesicle formation. Again, do not worry about the Clathrin-coated pits or the caveolae. Vesicular trafficking was the other type, and again, we're not talking about moving something from inside of the cell or moving to the outside or vice versa. We're talking about moving something within the cell from one area of the cell to another. So, for example, moving it from the endoplasmic reticulum to the Golgi. It's not actually leaving. Either way, it requires energy. And then the final thing we need to talk about is exocytosis, and that's the opposite of endocytosis. We're taking something that's from within the cell and basically ejecting it or spewing it outside of the cell. This can be activated by a bunch of cell surface signals or even changes in the membrane voltage, hormones, and neurotransmitters. Not so important, and definitely do not worry about your textbook goes into V snares and T snares. That's definitely too much information for A&P1. Just understand what exocytosis is, understand the three types of endocytosis, what they are and what the difference between those three are, and what vesicular trafficking is, and that all of those processes are a type of active transport and that they are going to require energy in the